also, after your initiative of moving to industry, they will be interested in that. So thanks a lot. And
thanks for inviting me.
You can share your screen and
yep.
Let's go for your presentation. Thank you.
Thank you so much, Stefano. It's a really huge pleasure to have you here with us.
Thanks for inviting me. It's a very big pleasure to be here and present today. I present myself as director of the scientific board for MirNat. And actually, I think I'm still a former scientific director of the company. But as I said previously, I officially left the academic environment just 3-4 months ago. And I moved to GSK as computational scientist for non-clinical safety department. But today I want to talk and speak and present about a huge research branch that we conducted together with Professor Vittorio Colizzi and many other researchers belonging to the MirNat company regarding the plant microRNAs and the cross-kingdom interactions as a cutting-edge technology for personalized medicine. So I have put my QR code here, so people that are interested can look at my profile later and look at the publications and so on if you're interested more on my profile. And please reach me by email if you have questions about the presentation or what I do later than the presentation a few days later. You know, sometimes questions come in mind later. So that's the presentation outline of today. It seems long, but it's not going to be very long. I'll do a brief introduction on microRNAs and their role in the regulation of cellular pathways. And I will bring a focus on the role of microRNAs in the cross-kingdom interaction. And I will also speak about the problem that is affecting the recent research about the fragmentation of data across the literature. So, and how, as a company, we decided to solve this problem regarding the microRNAs. I speak about later. Then I'll present few products that MirNat generated and created over the years from the foundation that was in 2012, and which are the future goals of our company in terms of research and in terms of delivering products that would help and guide the personalized medicine based on microRNAs. So microRNAs are small non-coding RNAs that are very small in size. So the maximum length is around 24 nucleotides. So the range is between 16 to 24 nucleotides. Their biogenesis is composed of many steps. So the biogenesis of microRNAs starts in the nucleus, where the polymerase RNA polymerase 2 generates an RNA molecule that has sometimes very complex secondary structure, which is called primary microRNA. Then this primary microRNA is processed by a protein complex composed by DROSHA and DCCR8, also called PASHA, that cleave the primary microRNA into a smaller molecule, which is called pre-microRNA. Then this pre-microRNA is exported outside of the nucleus in the cytoplasm by a protein that's called exportin 5, and then is processed in the cytoplasm, is processed by a protein complex guided by dicer, the dicer protein, that cleave this pre-microRNA, and the double strand is then cleaved again, and just the most stable RNA sequence is conserved and is acquired by the RISC complex composed of many proteins. And this shorter sequence is called mature microRNA. So this mature microRNA, together with the RISC complex, can regulate the post-transcriptional function of few mRNAs by cleavage of the target mRNAs. These happen in plants mainly, and by translational repression that happens mostly in animal species. So this categorization of the way of regulating gene expression in cells between plants and animals is crucial for our research. So keep in mind that the behavior of a microRNA in animals and in plants is different. And the role of microRNAs has been associated with many biological functions, with many pathways, and also with many diseases, cancer among all, because microRNAs regulate many pathways involved in, for example, the cell cycle arrest or the apoptosis. And so a deregulation of the microRNAs in the cell can lead to many alterations in pathways that are the basis of cancer development and progression. And if we look at the number of articles that speak or talk about microRNAs and several diseases like cancer or immune system or immunodeficiencies, diabetes or cardiovascular diseases, you see that from 2002 and in just 20 years of research, the number of publications increased a lot. So from just 10 to more than 10, or around 10,000 different publications. And as you can see also, the number of these papers is going to increase also from 2019, where the worldwide research was affected by COVID-19 pandemic. What about microRNAs and Crosskingdom? That's a new branch of research that actually is very new, because it's just 10 years old. And everything started when in 2012, Zhang and co-workers published on Cell Research a very interesting paper about the role of exogenous plant microRNA 168A that was extracted by from Oryza sativa, so the rice. That was able to specifically target mammalian, so mice, LDL-RAP1, which is a receptor for the cholesterol in the blood. And they essentially the experiment is the key figure of the experiment is in this slide. So these mice were fed with high quantities of rice, and after many days of diet enriched with rice, the level of microRNA 168A was increasing in the liver, and the increase of this microRNA 168A was related to a downregulation of LDL-RAP1. So that's an important point of this paper, was that the microRNA 168A, once it entered inside the mammalian cell for acting or for regulating the expression levels of LDL-RAP1, was acting as a mammalian microRNA. So the plant microRNA, once it entered inside the mammalian cell, it used the mammalian machinery for regulating the genes, and so is not by target mRNA cleavage, but by translational repression. Another key article that came out just 4 years later than the first article in 2016 regards the cross-kingdom inhibition of breast cancer growth by plant microRNA 159. So the starting point of this study was very easy. So essentially, they looked at the presence of exogenous microRNAs in the sera of Western donors, and they found that the level of plant microRNA 159 that was that had corresponding sequences in Arabidopsis thaliana and the mice and also the soybean. The abundance in serum of this microRNA is inversely correlated with the breast cancer incidence and progression in the patients that donated the donor. And this microRNA in the sera was detected in extracellular vesicles and was resistant to sodium periodate oxidation. So essentially, these microRNAs were resistant to degradation thanks to the presence of these extracellular vesicles. By transfecting cells, breast cancer cells with synthetic mimics of microRNA 159, researchers discovered that this microRNA was able to inhibit the proliferation of these cells by targeting TCF7. And so they created a model where microRNA 159 was able to downregulate or inhibit the expression TCF7, that is a downstream activator of MYC gene, and so the cell proliferation. So essentially, whenever the levels of microRNA 159 from plant was high, the cell proliferation decreased. When we started looking at the literature about microRNAs, we discovered that PubMed, especially PubMed, and the literature in general is a massive source of data. But the problem with this data is that it's very well fragmented. So if we plot the number of publications over the years together with the data extracted from GEO, talking about microRNAs over the same period, you see that there is a concordance of the trend. So whenever the publication increased, also the number of data increased. But most of these data are deposited into GEO, for example, which is the Gene Expression Omnibus, by research groups that are not affiliated to any consortia, like the Cancer Genome Atlas or ICGC, which is the International Cancer Genome Consortium. And so it's very difficult for researchers to retrieve this data and group this data according to the technology or the platform and so on, and reuse this data for other research purposes. So the challenge here is to identify a bioinformatics method that would allow to programmatically retrieve this data and the corresponding metadata, and also aggregate the data according to the same platform or integrate data coming from different platforms. So the aim of our research was to explore the microRNA-mediated cross-kingdom gene regulation, assuming that that's a very high-risky but promising research field. Why? First of all, when the first article came out about the cross-kingdom interaction of microRNA 168 with LDL-RAP1, there was a huge concern from the scientific community because of the reproducibility of the experiments and because of the impact of a discovery like that. So imagine that if the role of plant microRNAs becomes stronger and stronger and becomes get a statistical impact also on the community, probably we should add the microRNA molecules as molecules that are important for the diet. So not just proteins or lipids or sugars or carbohydrates, but also microRNAs can affect our body during due to the food intake. So there is an impact on the scientific side, but also on the economics. So there are big economic implications from this discovery. And so what we aim is to make a sustainable research, and I want to put a light on that because sustainable research is doing research with almost zero money impact. That's important. So we are in a period of time where everything is very unclear, especially with this COVID-19 pandemic. And so as a company, our main role and our main aim was to make a research that would be sustainable from the economic point of view. We binded a lot of scientific collaborations, especially for artificial intelligence for creating reliable prognostic tests or generation of new therapies. And we also created few industrial prototypes for scaling up the microRNA production, for creating functional food, and for generating cosmetics products. So in 2012, we founded MirNat, which originally was a startup company linked to the University of Rome Tor Vergata with the aim of performing research on microRNAs and Crosskingdom. Just a few years later, in 2016, we published our first research paper on microRNAs and Crosskingdom, and the computational biology was the starting point. So in 2016, we published MirCompare. MirCompare is the first dedicated algorithm for comparing plant and mammalian microRNAs. And this algorithm search for functional homologies. So what does it mean? Taking a set of plant microRNAs and a set of mammalian microRNAs, for example, human microRNAs, MirCompare identify the plant microRNAs that will have a putative role, a Crosskingdom role in the regulation of mammalian genes. By looking at homology sequence, not just homology sequence, but by searching for functional homologies among the sequences. So we based our research on a previous paper that was published by Brennecke and co-worker about the principles of microRNAs and target recognition. So in order for a microRNA to target an mRNA in the mammalian environment, we just need almost 48% overall sequence homology and 5 out of 7 homology in the seed region. So considering that the Zhang and co-worker declared that the plant microRNAs, once they enter in the mammalian cell, they start acting and using the mammalian machinery, then we can assume that the plant microRNAs behave as mammalian microRNAs once they enter inside the target cell. And so here I schematize what the software does essentially. So given a sequence pair for one plant microRNA and one mammalian microRNA, it first perform a global alignment and then a seed alignment, and then calculate the statistical significance of the alignment and of the functional homology between the two sequence pairs by making multiple comparisons over scrambled sequences that are generated by with the same percentage of A, C, G, and U for the mammalian microRNAs that is considered in the analysis. And in this way, we can also calculate a global p-value for the global alignment and a seed p-value for the seed alignment. And based on these two p-values, then we can filter the statistical significant comparisons and select the plant microRNAs that would act in a cross-kingdom interaction. We also experimentally validated MirCompare, and we focused the attention on two plants that are with a great economical potential. The first is Olea europea, which is olive from the European country. So why we decided to focus the attention on that? Because it's an essential component of the Mediterranean diet, and Italy is the first consumer in the world of olive and olive oil. So we selected from Olea europea 37 microRNAs. We didn't sequence the microRNAs from Olea europea, but we took from a previous publication. And that's another important bit. So speaking about making a company that is sustainable means also that data repurposing is has a key role on that. The second plant we focused the attention is Moringa oleifera. So this is a plant that became famous probably in the last 10 years in Western countries, at least, especially in USA and Europe. Is a plant that has been used for centuries in the African country, in the African continent, and is called the miracle tree. Is an entirely edible plant with very high nutritional values, has an anti-cancer and anti-inflammatory effect, and also can lower the cholesterol and sugar blood levels. Is used a lot in the traditional medicine in Africa. And the MirNat company was the first in sequencing and characterizing the MirNome, so the total amount of microRNAs in this plant. We identified 94 microRNAs conserved among other species, and also 2 novel microRNAs that are just of this plant. We use MirCompare for comparing the Olea europea microRNAs and the Moringa oleifera microRNAs with the Homo sapiens microRNAs. And we identified 3 microRNAs in Olea and 4 in Moringa that are capable or putatively capable of regulating the human genes. After making target predictions and enrichment analysis, we identified 1 gene, very important actually, that is a direct interactor of P53, which is called SIRT1, that is putatively regulated by these plant microRNAs. And we tested the role of these microRNA on several cell lines, Jurkat cell lines, THP1, and also PBMCs in order as a control, in order to identify the role or to validate, the role of this microRNA in SIRT1. Interestingly, but it's just a case that the microRNA is the 168, but the sequence is different from the 168 identified by Zhang and co-workers. And as you can see in the picture, both in Jurkat and THP1 cell lines, an upregulation of SIRT1, an upregulation of MIR 168 is able to downregulate the level of SIRT1. So we validated the role of these microRNAs from plants to downregulate the expression of SIRT1. And we published that. And we also patented the role of these microRNAs in in the regulation of SIRT1, and so a putative or a potential role in the treatment of leukemia. The next step was to maximize the production of this microRNA. Extracting microRNAs from plants is not the best way of doing in an industrial environment. There are many reasons. First of all, because the amount of microRNAs that you can produce or you can extract from the plants is minimal respect to the amount of the plant. So it's not it can be expensive, and also the growth of the plants is affected by the environment, and also depends on geographical or seasonal factors, especially for Moringa oleifera, which is an African plant that requires high temperature for growing. And so we decided to move from synthetic mimics, which is the alternative way for having high amount of microRNAs, to a natural mixture. So we, as a company, we focused the attention on creating something natural, not chemical. So the solution to this problem was to create or to grow the callus of the plants. So callus are an agglomerate of pluripotent stem cells from plants that normally appear in the plants, especially when you cut, for example, a branch. Then when you cut the branch, the callus appears for covering the cutted part, and this portion or this way for bending the plant is made by these pluripotent stem cells that have a huge potential of producing microRNAs. And so plating callus from, for example, Moringa oleifera is a promising bioproduction platform for these natural products. And you can grow them in a controlled environment and independent from geographical or environmental season factors. So in order to maximize the production of these microRNAs in the callus systems, we collaborated with the Laboratory of Botanics in the University of Rome, and we started stressing this callus with abiotic stress. And we identified as the best stress you can give to an African plant as the cold stress. So the cold stress applied to this callus from Moringa oleifera plant allow to maximize the production of plant-specific microRNAs that can be used for the cross-kingdom analysis and predictions. And after stressing the callus, then we resequenced we identified the microRNAs from this stressed callus with the low temperature, and we identified one specific microRNA that is called cold sensitive microRNA. So it's a microRNA that here we'll call just XYZ for patenting purposes, that is highly upregulated just when a cold stress is applied to the callus. And we tested, we looked at the regulation, at the pathways of this microRNA XYZ and how can regulate the functionality inside the cell, the plant cells. And we see that actually this microRNA is involved in the regulation of the cell growth and also in the maintenance of the meristemal identity and in many catabolic processes. So it makes sense for this microRNA to be upregulated when the callus is stressed with the temperature. So the presence of this microRNA in the plant cell inhibits the cell growth and inhibits all the catabolic processes in the cell that will lead to the replication of the cells. And this makes totally sense, because whenever the cell is stressed, the first thing that has to stop is the replication of itself. So the real question about this microRNA wasn't about the pathways that were regulated in the plant, but what about the cross-kingdom interaction of this small microRNA with the human. So in collaboration with Dr. Arianna Guidi, which has been a PhD student at the University of Rome, and started collaborating with MirNat in an industrial environment. So it was very helpful and useful for her as well to grow as a PhD student in an industrial environment. And together with Professor Roberto Bei, which is head of the Laboratory of Cancer Pathology in Rome as well, we tested the role of this microRNA in the regulation of the breast cancer growth. So together with, we used MirCompare for identifying the human genes that are involved in the cross-kingdom regulation. And we identified a set of genes that are crucial in the development of the breast cancer. So we performed an in vitro screening on breast cancer cell lines, and what I'm showing you here is an in vivo screening on xenograft mouse models. And as you can see, once the microRNA mixture extracted from the cold stress callus is given to the mice, to the xenograft mice, you can see that the tumor volume in these mice is the growth of the tumor volume is lower respect to the control or the unstressed callus. It means that this cold stress callus microRNA mixture contains specific microRNAs that have a role in the inhibition of the breast cancer cells in the xenograft mice. The next question is, how can we vehiculate these plant microRNAs in the human body? So it's very easy in mice, because then you can just do an injection in the site, especially for the xenograft mice, but you cannot use this way for humans. And when you want to make clinical trials, you have to identify a way, an effective way for vehiculate your drug into the human body. In this case, our plant microRNAs. And in collaboration with the Navetech, which is a spin-off in the University of Palermo, we engineered microvesicles from the orange peel. So this is 100% natural way of delivering the drug. So nothing chemical again. So the idea is to take our plant-derived microRNA mixture, vehiculate inside these microvesicles, and once these microvesicles containing the plant-derived microRNAs are ready, then these microvesicles can be acquired by the recipient cells in vitro, or can be given we can make tests in vivo. And so the last step would be doing clinical trials. So at the moment, we are in the stage that we are still engineering these microvesicles for vehiculate microRNAs, plant microRNAs inside these microvesicles. The last point of this presentation that I would like to put the light on is about data repurposing. So I told you many times that our aim as a company is to make sustainable research. It means zero impact in terms of money. Essentially, we want to minimize the amount of money for our research projects, not because we don't want to spend them, but because we think that the literature contains a huge amount of data that needs to be further explored. And most of the answers to our questions have been, and most of the data that would allow helping in answering our questions have been already generated. And so the data repurposing became our next generation business model. We took advantage of the publicly available data from the literature to reevaluate the Crosskingdom function on new microRNAs. And it's a strategy where bioinformatics has a central role. We can perform high throughput screenings of microRNAs with limited costs, and also we can perform multiple research projects at the same time by just using bioinformatics, and so making them 100% sustainable for a smaller company. We developed MAP, which is the microRNAs analysis portal that relies on another tool I published few years before that is called SMAC, the Smart Automatic Classification System. So MAP is a framework for exploring the microRNA focused literature and extract the molecular data associated with these papers. So the first step is to explore the literature focused on microRNAs and extract a series of metadata from the publication, together with, for example, the medical subjects that allow to categorize this paper according to the medical area, and the GEO datasets, so the gene expression omnibus datasets associated with these papers. Once we identify the association between the PubMed ID and the gene expression omnibus ID, then we can download the molecular data, and we can perform a series of bioinformatics analysis, starting from the quality control of the data, mapping array probes to the gene names, and identify, for example, cancer samples if the study is focused on cancer, and then perform cancer-specific analysis on this data. We can also infer interaction networks, identify genes, and we can identify genes that are mostly cited in these publications, and so the genes that are mostly relevant to the topic of microRNAs. These are a few statistics about the microRNAs analysis portal. So we collected and we explored more than 100,000 papers, and we categorized these papers in almost 92K different medical subjects. We also performed tons of bioinformatics analysis automatically on these papers and on the data associated to these papers. And some of these analyses are specific for cancer, like estimation of tumor purity or the molecular classification of the cancer, or in case of breast cancer studies, the detection of the receptor status. So this is not just about numbers. This is about our final scope as a company and as a research group, is to create or to empower artificial intelligence for the personalized medicine. So the amount of data that came out from the data repurposing made by MAP allowed to gather and to get an enormous amount of data focused on microRNAs, but not just microRNAs. We do have mRNA expression, so transcriptomics. We do have proteomics. We do have clinical informations. So we know most of the clinical outcome for many of these samples that we collected through the literature. And so we can use this huge amount of information for creating a training cohort, where we interconnect all these layers of information together with the previous knowledge about the endogenous down-regulated microRNAs versus the plant microRNAs for creating classification models, regression models, linear and non-linear regression models, so we can identify microRNA patterns that lead to longer survival respect to a control group. And so the final aim is to create to empower personalized medicine, and in particular for giving a mix of plant microRNAs for restoring the healthy condition given a specific disease. So to summarize the presentation of today, so the sentence is, we are what we eat. And this was coined in 2012 when Zhang and co-workers published this first paper about microRNA 168 from rice. And also just 4 years later, the plant microRNA 159 is associated with breast cancer incidence and progression. The plant microRNAs can resist to digestion, high temperatures, and also enzymatic action. They are detected in extracellular vesicles. And there are other papers that speak and talk about the role of plant microRNAs and their resistance to digestion. For example, the microRNA 172 was found in the stomach, intestine, serum, feces of mice that were fed with plant RNA extracts. So these microRNAs are very resistant. And as a company, our role and our mission is to elucidate the microRNA-mediated cross-kingdom interaction. So we produce many papers and we produce many products, but the key products here are MirCompare, which is the first algorithm for predicting plant microRNAs with cross-kingdom capability. We validated MirCompare in Ole Europea and Moringa oleifera, and we identified one microRNA able to regulate SIRT1 oncogene. And we also maximized the production of selected microRNAs in a total natural way. What's next? So we completing the study about the regulation of Moringa oleifera the role of Moringa oleifera microRNAs in regulating the growth of breast cancer cells. And we are working on finding a 100% natural way for vehiculating plant-derived microRNAs into host cells. We empowered, and we are continuing to empower, the data repurposing as our main business model in order to use artificial intelligence for personalized medicine. There are many people I want to thank. It's not just myself. I mean, I'm just a speaker. But this work that I presented today is made by many people that collaborated and give their own contribution to these projects. I want to personally thank Dr. Andrea Galgani, which is the director of the technical board in MirNat, and most of the experiments of validation has been done by himself together with Dr. Carla Montesano, Antonella Minutolo, and Marina Potestà, and many people at the University of Rome. And I want to thank you for your attention, and please make questions if you have any. Thank you very much.
Thank you so much, Stefano, for your outstanding talk. Wow, so many data. I think it was just inspiring. And what it was absolutely nice was also see how the integration of different fields can really change and can really be a huge challenge, right? Also for human health. So I think that actually this one was an absolutely fantastic example for all the students. I think that all of them, and I hope that they can really be inspired with your presentation. And I want to say that actually, so I think that they can really realize how important is nowadays the use of bioinformatics, how bioinformatics may be absolutely challenging, and how actually different fields, and I can say like this is actually also the results of a really well-structured research group, right? So it is important for all of them to know that actually we can make like the difference together, right? So I think that
the collaboration was crucial in this context because we had very different backgrounds, and yeah, I mean that was totally crucial, the integration of different expertise to reach the goal.
Absolutely. And actually, I think that is quite, so, you know, I was quite, it's just, I can say like a curiosity, to be honest, and then I can open for question, because also I am still in the escalation, you know, of a possible academic position. I was just curious why you decide like to move from the academy to the industry, and or as well as company, whatever, and how you think that actually these two different worlds, you know, they can come together in such way. So if you can just
I mean, regarding my you know, my personal experience, what Professor Colizzi, which is present, and I'm glad is present during this presentation, he always provided me the possibility to work in the academy together with the company. So consider that I started working for MirNat, and I founded MirNat when I finished my master. So I still haven't started my PhD when I founded MirNat with him and with Andrea Galgani. So I have done my whole PhD working half day during the PhD in the morning and the afternoon, and then late afternoon and the night for MirNat. So it was very challenging time. But actually, I feel that microRNAs was a kind of passion, so I didn't find it as a work. So it was a kind of, you know, very small bubble where I can give my own contribution, and I can provide my and gain experience on something new, and at that time was something truly brand new. So it was something that no one explored. So I was one of the first bioinformaticians working on microRNAs and Crosskingdom, and it was exciting. At some point, so regarding my very last decision to leave academy, so the academia, it was just because, you know, it can be hard to progress. It can be truly hard, probably too hard. And when you start, you know, having a family, or you know, your priorities slightly change. And so, but honestly speaking, I was very lucky, because even I joined a company, I don't feel the difference, honestly, because I'm working in a R&D department. So we do pure research. I have my own research projects, as I had at the UCL or at the Barnes Cancer Institute, and that's just research. So I don't feel the pressure of finding something for commercializing or something like that. It's not
like numbers, right? Yeah.
And regarding MirNat, I had officially had to step down, to make a step down from MirNat because of contract boundaries with GSK. But I'm always open to scientific discussions with a member of MirNat, and I always bring my expertise on microRNAs together with them. So it's not ended. It's not a kind of relationships that's ended. It's just a different shape. But we are still collaborating, and I'm still giving my providing support to many projects conducted by MirNat.
And this is actually absolutely amazing. This is why I say that actually it was absolutely clear how these two fields, these two work, they really can come together. So thank you for inspiring all of us.
Oh, thanks for inviting me.
I think that
No,
actually
it's a
the
pleasure.
Professor Gloria
Gloria,
Franco wanna
go
say
for it.
something. Yeah.
Hello, good afternoon, everybody. And thank you, Stefan, for a very nice presentation. Very interesting, amazing. Thank you. I have some issues, like more philosophy than science, actually. You are what you eat, you say that. And then when you have a vegetarian diet, you could be more influenced by the microRNAs from plants than microRNAs from animals. And do you think that we can change the status of diseases depending on being more vegetarian than carnivorous? This is one question, but okay, one issue, and the other one I can ask you after.
Yeah, I mean, that's a very interesting point, and thanks for spot the light on that. So actually, yeah, the short answer is yes, because what Zhang and co-worker identified is that the levels of microRNAs in the sera increased after a long exposure to that specific diet. So speaking about a human, if you eat, for example, rice once a week, you don't increase your amount of microRNAs from rice in a level that allow you to have a regulation. You should eat rice every day. For example, this is that there is actually a study that is I think is already published about the diet based on rice for the Chinese population or the Asian population, where the rice is the main source of diet in their case. In that case, that would help. The purpose of our study, more than using the pure diet as a regulator for the gene expression, is more to identify the regulation mechanism. And then once we identify the actor of the regulation, then we can provide integrative or diet support for, if we identify that one microRNA from a plant has an active role in regulating one gene, then we don't have to eat that plant for forever, but we can just extract that microRNA and use as a food supplement for reaching the levels that we need. That's the most important thing. But the diet has a central role. And that's the reason why we focused on olive oil, because Italian people and European people use a lot the olive oil. It's the central of one of the central food of our diet.
And the other one is, when you say about the targets of the microRNAs from rice and from olive oil or other plants, you are looking at the human point of view. But the plants produce the microRNAs to control their metabolism, not the human metabolism. What kind of similarities you have in the targets for the microRNAs in plants and in humans? Because they are related to oncogenes or cholesterol metabolism, but this is typical from mammalians. What happens in the plants?
We have conducted a research on that. So prior to look at the regulation in the mammalian genes from plant microRNAs, we looked at the regulation of plant genes from plant microRNAs. And actually, it's interesting because the microRNA 168 is from Moringa oleifera, for example, is one of the main regulator of the cellular, the proliferation of the cells, which is exactly, and the regulator in the human counterpart has exactly the same function. So in some way, the microRNA from plant has a role in regulating the proliferation of itself in case of stress conditions, but it's actually the same function that you would find in the human cell. This is not always true. mostly because there are some functions that are peculiar of the plants. So you don't have a function, a counterpart in the.
A internet desligou?
Sorry?
Sorry,
okay.
I don't know if it was my internet or yours. My connection is unstable. Probably.
Can you hear me?
Probably.
Yes, now,
Yes,
now I can hear you.
I was just saying that sometimes the micronase from plants don't have regulate genes just in plants, but they don't have a counterpart in the human or in the mammalian, just because these are peculiar functions of the plant. But actually, yes. And there is also a very interesting paper I didn't flag in the presentation about the interaction between plants, a flower, I don't remember the name of the flower, and the bees. So it's not just human, but also other animals or insects use microRNAs for their own regulation of functionalities. So plants help us, but we don't help them.
Thank you so much. Thank you for the explanation, for a wonderful seminar.
Thank you.
And okay, thank you. Hope to see you in Brazil.
Sure. We have some questions in the chat. I don't know if you can reach the chat from there. Also, if Juan Carlos wants to open the microphone and ask the question. João de Almeida also has a question. If you'd like to open the microphone and ask, you are welcome, guys. Or we can read.
I can answer to the questions if they. Regarding the question of João de Almeida, yes, the 48% sequence similarity and the 5 out of 7 is referring to the direct mature microRNAs. Yes. And we are conducting a study in collaboration with the University of Edinburgh about not just the pre-mature or the primary but also by looking at how the secondary structure affects the selection of the microRNAs, of the mature microRNAs, and how the pre-microRNAs can act in the cell if they are transfected. So how the secondary structure affects the behavior of the plant microRNAs in the human or the mammalian machinery.
And also, after your prediction, you tested experimentally, right?
Yes. Yes.
What's the percentage of success against fail? When you predict it's nice, then you test and it doesn't work, or it works?
Well, I mean, in the case of the Ole Europea and Moringa oleifera, we used a very strict way for filtering the predictions. So we just found 4 microRNAs from both plants, and just 1 out of 4 worked properly. It's 25% success. But on the other side, testing 4 microRNAs out of 1,000 has a cost, experimental cost, that's much cheaper respect to screening all wet lab based.
Sure. Juan Carlos question, let's see.
Yeah. First, maybe toxic if not handled correctly. Yeah, but okay. So regarding the first, would best be sufficient to test the effect in mice or human? Yeah, that's the point that also we discussed with Professor Gloria Franco. So we are not aiming at giving tons and tons of Moringa oleifera to the people for reaching the levels of microRNAs that are sufficient for the regulation. The aim is to identify the microRNAs from Moringa oleifera that are crucial for the regulation of mammalian cells. And once we identify them, we deliver them synthetically. So we deliver them not by providing the food, but providing a food supplement based or enriched with selected microRNAs. So in this way, the safety of that would be preserved.
How do you synthesize the microRNA?
No, it's a microRNA mixture directly extracted from the callus. So essentially, we know that the microRNAs, we know that the callus with abiotic stress, so with cold stress, contains a huge proportion of one microRNase over the other. So we can, let's say, is 95% of one selected microRNase and 5% of all the others. So, but we don't provide one synthetic mimic. We provide the mixture of the callus from moringa. So it's 100 natural product. It's nothing, nothing synthetic, and is delivered by microvesicles from orange peel. So again, it's nothing synthetic. So all the microvesicles are extracted from orange. So there is nothing synthetic. We don't buy the mimics for testing the.
But do you see some advantages of changing a little bit the sequence for the sequence to adapt to the human target better than the plant one?
It depends. It depends because whenever you change the sequence, first of all, you lose the patenting loyalty on that. And that's the first point. And that's the first point. The second point is that then you automatically is not a natural supplement, but is a drug. So whenever you synthetically change the sequence of the product you're providing to the animal, you're not giving a natural product, but you're providing something synthetic. So it's a different also procedure of approving of the type of treat. So it becomes a treatment and not a food supplement. But the functionality, actually, we have seen that the functionality of the pure plant microRNAs without any editing was enough for creating prototypes of food supplement. We are in the phase of producing prototypes, and the direction seems very promising. But of course, we are open to changes if we find it's not working as expected. Regarding the second question of Juan Carlos, do you think your research is a way to explain why vegan or crudivorous habits have good impact on people? I mean, that's a very bold sentence. I wouldn't say that, but just because the amount of probably that would be true for people that follow a vegan or crudivorous diet for a long time. Long time means years. If someone starts having a vegan diet or a crudivorous diet for, let's say, 2 weeks, 3 weeks, or few months, then the levels of microRNAs in the sera wouldn't be enough for having a strong regulation. But of course, there are indications of that. So our research is probably flagging the importance of having, we always say when you, one of the old sentences, if you eat apples or if you eat a lot of vegetables, then you are healthy. But the molecular explanation of that can be also in the microRNAs. So definitely we are in that direction. More healthy you eat, more plants or vegetables you eat, and more beneficial you can have from exogenous molecules.
So you think that the difference in the efficiency of extraction from the callus or people eating the plant is quite so big that would not justify some agronomists to cross the rices and find a new kind of rice that would produce a lot of that microRNA, for instance?
Yes. At the moment, we are not focusing on the different, species of rice, for example. So we don't know if the basmati rice has more micronase respect to another. But I guess that the effort from this agronomist that is partially economical, but is also partial, probably they're trying to maximizing the nutritional values of this type of rice.
Sure.
Has an implication, I guess, on the micronase as well, but we haven't explored it yet.
But the amount of microRNA that you deliver doing the extraction from kalo, I guess it's kind of in the same range, or like thousands of times different?
Respect to the natural diet, you mean? Respect to the normal amount of food, you mean?
Yeah.
Eating
the vegetable or delivering?
No, it's much higher. No, it's much higher.
Quite
yes.
a lot, much higher.
Probably 100 times more.
100 times. Okay. So
yeah.
That's the way to go.
Yeah, There was an interesting paper that was actually discussed a lot in the literature. It was published also on a very high impacting journal, but was about the role of micronase from red grape. And so the final sentence was that more wine you drink, more micronase you get, and this micronase may help you in several functions. But the quantity of wine that you should intake for reaching the levels of micronase that would get you drunk probably 24 hours, 7 days a week. So the beneficial role of micronase is totally reduced by the amount of the wine. So that's true also for other type of food. Also the Zhang and co-worker paper, they fed the mice with tons of rice, so they forced the diet a lot, which is not feasible for humans. So the focus of the company is not about one possibility is to reach in that food with the same micronase. For example, giving rice with the micronase from rice, but a higher quantity of micronase.
Dr. Vittorio, would you like to comment something?
It's very difficult because of what I'm just thinking, that we are losing a lot of time to do what Stefano is suggesting. Unfortunately, the economic difficulties and I am a university professor for more than 40 years. All best students go out of Italy. They go in the States, in England, in Brazil, Marta, for example, everywhere. This is really a problem. So when I touch with the hand all this possibility that we have with our students is really a pity. But this is life, and it's important that anyway science is developing everywhere in our planet.
Sure. Okay, Stefano, Dr. Vittorio, thanks a lot for your conference. I think it was very inspiring. Mostly also because you were showing how the industry works and the sustainable research. That was really great for our kids to think about. And
so if anyone has questions later than the presentation, also a few days later, please contact me by email, and I'll be happy to answer to any question or have a chat or whatever. I'm very happy to do that.
I want to say more. If somebody will be interested to participate at this kind of research or in this field, we are completely open. So maybe a good idea. So if we can start just for discussion, theory, data, vision, also to start to grow up. So this is not a closed startup. We have a lot of people which is joining.
Yeah,
also.
we actually also since you made the cross, we are also have some projects open with COVID-19 and plant micronase and Crosskingdom. So if anyone is interested in
Stefano, post your email in the chat here.
Of course, yes.
So I can distribute also, but
yeah.
Then they can have it. All right. So thanks a lot. It was a very nice afternoon here in Brazil. So have a good evening there. Thanks a lot for being with us.
Thank you. Thank you.
Thank you so much. Thank you so much.
Thank you, Marta.
It has been absolutely nice. Thank you, Prof.
Also for me. Take care, everyone. Bye.
Bye bye. Buongiorno. No, come si dice?
Arrivederci.
Arrivederci. Avanti.